Satenda, if you can just open your video and we can start. Thank you.
Okay. Okay, good morning, everyone. So today's presentation is going to be delivered. Okay, let me move my device slightly so that you are able to see me. So today's presentation is going to be delivered by Professor Matthew Gruwell. So Matthew is an associate professor of genetics and evolutionary biology at Pennsylvania State Behrend. Matthew also has a bachelor's in zoology from Brigham Young University and a joint PhD in entomology and organismic and evolutionary biology from UM. All right. He did a 2-year postdoctoral research fellowship at the University of Buffalo in phylogenetics and parasite biology. And then Matthew has been with Penn State Behrend for the past 15 years, where he was the biology department chair for the last 6 years. And currently, he was on sabbatical in Bangkok, Thailand, as early as in 2017, and currently he's on sabbatical at the University of Mpumalanga since October last year, and he'll be returning back to the US in June 2024. So his research has been focused on the evolution and systematics of endosymbiotic bacteria of scale insects. A few years ago, he started working on evolutionary DNA of the invasive species in North America as Great Lakes, and this research was funded with a grant from the Great Lakes Protection Fund. His work at UMP and with the Rivers of Life and the Aquatic Systems Research Group is looking at using eDNA to track the invasive red claw crayfish distribution, the distribution of tigerfish, and also looking at the distribution of tigerfish and the species overlaps that might exist using the yellowfish as a target organism. So I present to you Professor Matthew Gruwell for today's presentation. So the title of the
sorry.
Over to you.
Thanks so much, Satenda. Let me go full screen here. All right. I am really grateful to be here today. I'm really actually super grateful to be in South Africa right now. As Satenda mentioned, had an opportunity for sabbatical coming up. My real goal was to try and make it to South Africa and try and use some of the research skills I've gained with environmental DNA over the past few years to work with some awesome researchers down here. And I got really lucky as I was searching and trying to find people. I came across Satenda, and he was extremely welcoming. And we started talking about potential projects with the Aquatic Systems Research Group. And he introduced me to Gordon O'Brien with Rivers of Life. And so I'm just really, really grateful to be here. I'm going to talk about some of my work from the North American Great Lakes and from some of the areas in Mpumalanga that we're working. And my little short title to try and be funny is eDNA from PA to ZA, PA being Pennsylvania, where I live, way up here in the Great Lakes, or right on the Great Lakes, all the way down to South Africa. So I'm just really excited to be here. Just at the top of this, I want to shout out some great colleagues and collaborators. Ivor, Gordon, Tatenda, and Allegra are my co-PIs, and pretty much I ride their coattails all the time, and I'm very lucky to have association with them. And then some amazing students that we've interacted with and worked with from Amanda and Angelica, Annalise, Shedron, Ellen, Emily, Kyle, Noel, Abigail, and Ryan. And that's kind of the tip of the iceberg with some of the other good students that we have. A lot of great collaborators. I can't really, I don't want to sit and read all of them, but they're extremely beneficial and help our research in lots of ways. And of course, I want to shout out the Great Lakes Protection Fund, UMP, Rivers of Life, Aquatic Systems Research Group, Penn State Behrend, where I'm from, and a few other great organizations around Pennsylvania. So as Fatenda mentioned, I started catching bugs in high school, I guess metric, just before my metric year to go with South Africa terms. And I just decided, I always wanted to be a scientist from the time I was young, and I decided entomology was going to be my route. And so I did a bachelor's in zoology and a PhD in entomology. And for a long time, my research really focused on the evolution and systematics of insects and the bacteria that live inside them. I don't want to spend too much time going on it, but there's some great questions you can ask about coevolution. And so I was asking questions of armored scale insects and their obligate bacteria endosymbionts, and do they evolve together? Do they pass them on strictly? And this could be conversations for another day, but it's really, really fun, interesting stuff that I do hope to continue with and get back into someday. We also worked on Cardinium in armored scales, which is like Wolbachia. It's a sex ratio distorting bacterium. And we were able to show there's being passed between parasitoid wasps and the armored scale insects, which through horizontal gene transfer, horizontal transfer of the bacteria, which is pretty cool. I did some work on mealybugs as well, and their endosymbionts, and we're going to get into it, and some fluorescence microscopy showing the endosymbionts inside of the insects and where they're located. Just a few different random things. But then I got incredibly lucky in 2017, I believe it was. My good friend Ivor and colleague invited me to come be a part of a research project with him with the Great Waters Research Collaborative, where we would try and use eDNA or environmental DNA tools to confirm the presence absence of what they were finding with microscopy and looking through ballast water samples. And so I'll get back into this in a second. I want to just take a minute to go through a little bit of what eDNA is and how it's used and how we collect the data, and then kind of set the stage for the different projects that I want to talk about. And Ivor actually kind of spearheaded some of this stuff way back in the 80s when they were looking for ways to isolate nucleic acids from aquatic environments. And he came up with some of the very early protocols that would then go on to serve us really, really well now. Back then, we didn't have GenBank and the barcode of life. We didn't have all the tools and all the data that we have, the billions and billions of sequences that we have to look at now to compare and figure out what things are. But we stand on the shoulders of giants, and it's work like that that helped this stuff really take off early on. So what is eDNA? What is environmental DNA? Well, we are all sloughing off cells constantly. We're losing our DNA all the time. And it's everywhere. Everything we touch, everywhere we go, we leave a little bit of ourselves behind, a little bit of our code. And we can take that now in the environment, in water or in soil and other places, and we can extract the DNA out of that and learn what organisms are there because they're all unique. As you combine eDNA with DNA barcoding, we can get a good picture of what life is like or what life is there without maybe as rigorous as difficult sampling. I'll kind of get into that. So how do we do it? Well, it's pretty simple. First, you just collect water from the area of interest, wherever you might be going, and you need to try and understand maybe what's in there. If you're looking for an invader, you want to detect that invader, or you're trying to get a biodiversity sample, lots of things you can do for that. We filter it out, try and take the DNA out of it. Now you're not actually taking the DNA out. The filters wouldn't really catch just straight up DNA. It's pretty small, but the cells of those organisms that are there are going to provide you some DNA for your analysis. You extract that, so you put it through a process where you can get rid of all the proteins and the RNA and the cell membranes and other things like that, and leave you with just genomic DNA. And now you've got a purified eDNA sample. And in that sample, you'll have all the DNA of organisms that are there, not just the thing you care about, but everything else, because at this stage, there's no way to kind of separate that out. And that's where this next step comes in play. So now you take your eDNA samples and use PCR chemistry, and you create species-specific primers, or we can also call them probes, that will then find your target DNA sequence. We've used cytochrome oxidase 1 for the most part, because that's kind of been identified as a DNA barcoding gene. And there's the really great databases full of information on CO1, but you could actually use other genes as well. If there's more data on your target organism, by all means, use that. But it is nice to add to the barcoding literature of CO1 for organisms that you have. So those primers specifically land only on the DNA of the organism you're looking at. You can run them in a PCR. And if you have the ability, run them as a qPCR. And I'll talk about the difference between those for looking at detection. So if you run on regular PCR, you can run out through agarose gel electrophoresis, and positive bands are showing that you have the DNA of the organism you're looking for. And it's a great way to tell, but it's also can lead, it's an extra step, and it can add sometimes contamination issues. So you got to be really careful with that. Another option is using qPCR, in which case you can use these double-stranded DNA binding dyes to bind onto it. And then as it's read in this machine here, this Behrend machine, it actually can read how much of those binding dyes are attaching to amplified DNA as the process goes on, and quantify your PCR as it's going. So how does that work? You have your typical PCR profile here. As we go in minutes, we have your high peak, your denaturation peak using 95C, then annealing temperature, and then your extension step, and that's where those dyes are gonna bind onto your DNA. And as they bind, as the DNA starts to amplify and double, you kind of hit a crucial point of exponential growth in your PCR reaction. And the machine then can detect that and detect the amount of dyes that are binding to DNA as you're amplifying it and making billions of copies of your DNA. And then you want to make sure you're doing the right thing. You're amplifying the correct organism. So you can go through the melting analysis. So at the end of your thermocycling, the machine will slowly raise the temperature. And as it does that, it denatures your DNA. From that last slide, you have your double-stranded DNA, you've added all those binding dyes to, and then as it drops off, as you increase the temperature, it rips the DNA apart again, denatures it at a certain temperature, depending on the size of your DNA sample and the GC content and other things like that. And the dyes come off, and then you get a melting curve here that tells you whether or not you have the right thing. And so ideally you would run the process, amplify the DNA, sequence it to make sure you are doing the organism you think you are doing. And then after that, you don't have to do the gel. You don't have to look at it. You don't have to sequence it, because the melt curve can tell you if you've got the right thing. So here's an example of one of our assays. This is a serial dilution of an organism. And you can see here, each dilution is a 10-fold dilution. And that should correlate to a three cycle drop each time as we look at our CQ values. And of course, our melt curve here looks really, really solid. And so we have really good confidence in this assay that we are amplifying the organism we want to amplify. And then, we can look for presence or absence maybe of your invasive species. So let's go back to that first project. It was focused around ballast water. So the Great Lakes in the United States are one of the best resources we have, arguably maybe the best resource, because of the need of fresh water for everything. They hold about 20% of the world's fresh water just in the Great Lakes, and they actually act as a very slow river, where all the tributaries and everything that dump into Lake Superior and the other lakes, it slowly travels down through Huron and through Erie and Ontario, and then out the St. Lawrence River. So it's this amazing natural resource of water, but it's also a really important shipping resource because it's so inexpensive to ship things compared to rail or other methods. And a lot of the natural resources that need to be shipped are actually up in Minnesota, Wisconsin, that get on ships in Lake Superior and then are shipped down to the lower Great Lakes, where they can then get put on trains and other things, maybe in Toronto, Buffalo, Cleveland, Detroit, and these areas to go out to different parts of the country, or maybe shipped even all the way out. The problem is when you unload a big ship, a big Laker ship, you have to add a bunch of water to it to balance it out. And so they suck up a bunch of ballast water, and then they go back up to Lake Superior to get reloaded back on, at which time they have to purge all that ballast water. And as you can see here, here's the problem. You're going against the stream, against the river, and taking organisms that might be established down here up to the upper Great Lakes, and then possibly causing the spread of invasive species. Not to mention, just having shipping containers coming in and out all the time also can bring invasives from other parts of the world to this great resource that we have. And we have lots of issues with invasive species that most people have heard about, such as zebra mussels and other things like that. So don't need to get any data off of this, but what our essential problem we're asking is, as we look at uptake samples, discharge samples, source water samples, does eDNA work as well as microscopy and just searching for the organisms that might be there? And essentially what we found, I'm just going to give some key takeaways. We just resubmitted this after some revisions to the Journal of Great Lakes Research. So we're kind of waiting to hear, but hopefully soon it'll be out. But here are the major takeaways from this project. We wanted to look at eDNA as an early detection method for AIS. Now, I'm going to say AIS. In the States and a few other places I've been, AIS typically we use to reference aquatic invasive species. And I know from reading things here and talking to other people, typically in South Africa, AIS means alien or invasive species. So I apologize, but when I'm talking AIS, we're looking at aquatic invasive species. So can we get early detection of aquatic invasive species spread through qPCR? Can we get a good working protocol of qPCR for early detection of Hemimysis anomala, the bloody red shrimp? I've got some images of that later we'll talk about. Compare the eDNA traditional sampling to microscopic analysis. When we did that, our results matched really, really well with those that were found to have, found through microscopy, to have the organisms present. And in fact, we found successful eDNA detection at 0.2 organisms per cubic meter versus traditionally sampling, which might require 200 to 2,000 organisms per cubic meter. So very effective tool, and much more time, much less time consuming, much easier on our time. As part of this, we had some awesome students that went out looking for Hemimysis. It had not yet been discovered in the Pennsylvania waters of Lake Erie, and they found it, and it made quite a stir. It had been found in Lake Erie already, so we were fairly certain it would be there, but it had been searched for and not found. And 2 undergraduates and a grad student, just awesome, went out and found it themselves. So that was pretty exciting, made a big buzz for a little while with these guys. So after this project, Ivor Knight and Allegra invited me to work with them on a grant through the Great Lakes Protection Fund. And we had some number of targets we're trying to go for. We wanted to develop 5 working protocols and probes, or protocols and probes for 5 target organisms, being invasive species. We want to conduct mesocosmic experiments to measure the rate of eDNA signal. And those are the 2 big projects I'm going to talk about today. We're going to develop harbor monitoring protocols, validate eDNA probes, construct some and follow some QA/QC project didactic framework, and work with some stakeholders and do a lot of outreach with this project. So the next big thing was they'll talk about the quantitative assessment of planktonic AIS eDNA signal duration in Great Lakes Harbor water microorganisms. So this is kind of number 2 on our objectives for the grant. And the premise here is in the context of harbor monitoring, is eDNA methodology is useful for preliminary indication that will guide sampling and confirm presence of organisms. Or put a better way, Ivor came up with this, I really kind of like it. It's the presence of eDNA evidence. Presence of eDNA evidence is that evidence of organism presence. So is presence of evidence, evidence of presence. And then looking at the duration of the eDNA signal in the absence of live organisms. So our 2 targets to begin with for this are Hemimysis anomala and Daphnia lumholtzi. Dr. Aaron Gray from University of Maine developed working primers and good protocols which you kind of tweaked and made work really well for our lab for using Hemimysis anomala. And then we created some for Daphnia lumholtzi. And one thing to note from this, well, when we used cytochrome oxidase 1 for both of those, but you can see here, orange is Hemimysis anomala sightings and established spots, and light blue is Daphnia lumholtzi. Neither of them are very well established in Lake Superior, so it's kind of critical. We don't take these things up further north. And we'll talk about Cercopagus pangoi a little bit later, but we're going to focus on these 2 for this microcosm experiment. And we did this with using qPCR protocols that we developed. So here, one of the cool things, I don't want to get too deep in the woods. I want to have time to talk about a bunch of other things, but using techniques people come up with the genomic DNA and G-block analysis, we can actually quantify how many copies of the DNA are in a microliter of the sample. And that's really a powerful tool. So when we do these serial dilutions and then do the G-block analysis, it can help us do that. So here's some of our data for anomala and lumholtzi. And just to kind of get through the experiment, we took a bunch of the organisms, beat them overnight in a cool temperature to let them really kind of explode and break up, and then treated. And then we had a controlled treatment and a regular treatment. We're also testing raw lake water versus filtered water, but for time's sake, I'm not going to delimit or spend a lot of time talking about the difference between those, but we did have controls for both. And essentially what we found, as you can see here, this is elapsed time in hours. We carried this out for 70 days, but we knew after a few days, most of the signal would be gone. So even though we kept testing over a few months, we were at that point, we're testing once a week and once every other week. But in the beginning, we were testing every 12 hours. Shortly after that, every 24 hours and every couple of days and 2 or 3 times a week. But what we found here, as you can see here, 100 hours, about 4 days into it, we'd gone from 10,000 copies down to 10 copies. So within 4 days, almost all the DNA had been taken out of the system. We were not finding it. And within 150 hours, so you're looking at, maybe 5 days, we're looking at no amplification or below detection limit. So DNA is going out pretty quickly. Did the same thing looking at Daphnia lumholtzi. Again, 3 tanks with the organisms, 1 without, using lake water, using temperatures that we would expect to be in the lake during the summer months. We ran this one for only 10 days for good reason. You can see here within about a day, we'd gone from 500 copies to lower than 5 copies of DNA, and then below detection limit within 2 days. So lumholtzi went out even much more quickly, break up a lot easier, get chewed up a lot easier than the bloody red shrimp, though they're not that big, they're much larger than a Daphnia. And these data follow those which are found with removal experiments where someone's put maybe a fish into a system, allowed for some time for cells to maybe slough off and then taken out. Obviously, a fish sitting in water is going to take a lot longer to completely decompose and get rid of all the DNA than small little invertebrate organisms, but the removal experiments show very similar data to what we are finding. And there's good evidence that bacteria love to eat this nutrient-rich source, full of carbon and nitrogen and all these other stuff in the DNA. It's going to get chewed up really, really fast. So it's not a surprise that these the DNA dissipates really quickly. So the take-home from that is really exciting. The presence of evidence is evidence of presence. If we are getting DNA signal, the organism was likely there and live very recently. So it's, you know, if it's an invasive, maybe cause for concern, but it's a good tool. It works. I want to move on to creating probes for other invasive species that we looked at, right? This is another part of our project, create probes and working protocols for 5 Great Lakes target organisms. We're writing this up now. I know it's on my desk right now, and I'm working on hopefully wrapping that up as quickly as I can. But can we use eDNA as a tool for finding other aquatic invasives? So I won't talk much more about the bloody red shrimp. It has been a problem in the Great Lakes for some time. It came over from the Ponto-Caspian region. We find a lot of that happening. I think those lakes mimic well what's going on, you know, climate-wise and other things with the Great Lakes, and there's a lot of shipping going on between them. So we get a lot of invasives coming from there. The probe developed for Daphne lumholtzi. To do this, I'll just kind of spend a little time on this one and then maybe go a little more quickly through the other ones. You need to look at distribution, geography, morphology, and phylogenetics to make good probes. You want to find things that are closely related that you can test against if you can, because you want to make sure you're not getting signal from a different organism. That can be really problematic. For Daphne lumholtzi, Daphne lumholtzi, we were looking at Daphne magna as a test. They're fairly closely related to each other, and they sometimes overlap in where they live. And it was readily available. We had the organisms, which is kind of nice because we want to test them against Daphne lumholtzi. So here we have the spiny the African spiny water flea is what I've read it as being called. It's from East Africa areas, from what I understand, and it's also from Asia. I'm not sure we know exactly where it came from, but it is becoming an established aquatic invasive species in the Great Lakes. It's actually more of a problem in some of these other areas near the Great Lakes, and we're worried about getting into the Great Lakes because all rivers kind of flow that direction in this area. They don't go south to the Gulf of Mexico. So here's some of our tests using agarose gel electrophoresis. Again, lumholtzi, exact same primer, exact same protocols with Daphne magna, no amplification. And we have the exact opposite figure as well, with the magna primers working beautifully and lumholtzi primers not working at all. So a successful probe. We've already looked at our G block. Here's our DNA quantification analysis. Again, CQ values can't predict really well the log 10 copies of the DNA we're looking for with Daphnia. So really helpful. We made some for Cercopagus pangoi. Again, another invader from the Caspian region. Looked at its closest relative, the Bryotrepes longimanus, which actually is also found in the Great Lakes. Looks like we just went through a little load shedding, but we're back. Bryotrepes is even more all over the Great Lakes. Cercopagus over here to the left, not so much yet. Hopefully we can help some early detection. Here's the large one, Bryotrepes versus Cercopagus. Similar to Cercopagus, a lot smaller. And they call it the fishhook water flea because of this end here. And it ends up on fishermen's lines all the time. Here's our protocols. Won't worry about those. And our standard curves. Just want to get this for time. We really want to do a plant. We decided on starry stonewort, which technically is kind of in that area between plants and algae. But we went with Nidolopsis obtusa, which is becoming a big problem in the Great Lakes and other lakes in North America. Again, looked at some phylogenetic evidence and found species as closely related to, developed primers They seem to be working really, really well. Here's our qPCR data for that. These things have a much higher detection limit than others. They don't shed off nearly as many cells, it seems, than your animals do. It's kind of interesting. And we also didn't use CO1 for this, of course. We used MAC-K as a classic plant barcoding gene. And here's our curves for that. I would like to get into the scuds. The scuds have been a little bit of a nightmare for us. Luckily, we have one student, Abby, who has wanted to become a scud pro, and she's still trying to work it out. We wanted to go for Gammarus tigrinus. Apparently, that is all over the Great Lakes, but we actually could not find it. We had a lot of difficulty finding Gammarus tigrinus. And then as we dug into the literature and into the barcoding data, it seems to be kind of a mess. So then we switched to Echinogammarus ischius, which we could find, but again, taxonomy and barcoding and other things are becoming kind of problematic. In Europe, it's called Chaetogammarus ischius. It seems the exact same species as Echogammarus ischius in North America, so that needs to be sorted out quickly. Seems the European scheme is probably right, because here it is in a phylogeny versus all these echinogammarids down here. So we're still working on good probes. We have some that work, but our standard curves and our dilution series haven't come out perfect yet. So that's kind of a project I may or may not tackle upon returning. So I want to move to some of our South Africa projects, because these are probably the most important and of the most interest. Like I said, Tatenda and Gordon have just been amazing hosts. They've helped me out in a million ways. I've gotten to do lots of fun field work in Kruger and other places around in Mpumalanga. And I'm just having the time of my life being in the most beautiful place on the planet and amongst the coolest organisms and coolest people. I just can't really say enough great things about my time here in South Africa. I just wish I could spend another year. So we have some eDNA projects that we're working on. eDNA detection of red claw and red swamp crayfish. Tools for tracking tigerfish and eels, like Tatenda kind of mentioned. Tools for determining species range of yellowfish in the Sabi River. And then some meta-barcoding projects we want to get to that will probably be collecting data in the next couple of months, and hopefully I'll be able to analyze, after returning to the States in June. So the crayfish, we know these are a big problem, especially the red claw crayfish, huge problem across South Africa. We've developed probes, and I've got the progress on this right now. We've got genomic DNA, we've got water samples known to have DNA in them from places that have the red claw crayfish already. And then I'm hoping to go out with Shedron soon, and we can collect all through Kruger, get a bunch of water samples, and learn more about the distribution in the lower Kruger, at least, of red claw crayfish. For that one, we have DNA sequence for CO1, so it's easy to make primers. We've made primers, and I'm testing those as we speak. We've got DNA extractions going, and we're about the same place. We were able to get a DNA sample for red swamp crayfish. Luckily, it is not nearly the problem that red claw crayfish is right now. There is one location in Mpumalanga that have been noted from, and we've got water samples from there. And I was able to go out in the field and see them and confirm their presence. Unfortunately, they're still there in I can't remember how to say the farm, Dry Hoke Farm. Hopefully they're not extended beyond that. We're going to take some samples all up and down the Crocodile River to see if they've kind of left that area very far. And hopefully we can get this up and working. With tigerfish, we really want to look at kind of distribution. So the Rivers of Life Research Group have made these amazing hypothesized distribution maps for a whole bunch of different fish, looking at tolerance, taking all sorts of different factors into consideration on different rivers all across Mpumalanga, focusing on Sabi, Crocodile, and Kamadi rivers and those drainages, and made hypothesized where these organisms should be. So hopefully with eDNA tools, we can look a little bit at the distributions of tigerfish and of some others. Tigerfish, turns out Hydrocyanus vittatus is quite tricky because if you look at the phylogeny of these things, you can see there's a massive amount of variation in vittatus. And so as I pulled in all the different sequences of CO1 that I could find, they're all different. There's a lot of difference within the species. And I'm not a fish biologist. I probably won't be able to work that out and see if there's multiple species or not. I'll leave that to Sandy maybe and her team, some people that are better than I am at this, some of the fish stuff. But finding primers that would kind of hit any one of the populations is tough. So we have we're working on testing primers and protocols now. I'm just trying to get some more samples and some more, hopefully water samples. And then hopefully we can sample on the Sabi some more. We've got some good sampling on the Sabi River already and also on the Crocodile River coming up. Eels are I'm just kind of dipping my toe in this. I know even less about eels than I do about fish probably. But we want to do the same sorts of things because I think there's less known about the distribution. And again, we have all these awesome hypothesized distribution maps of where they should and could be. And so hopefully these tools will work out, trying to get some good DNA from them and then get some protocols working. These actually present the opposite problem of the tigerfish. They're actually extremely similar across the 4 species that are found in this part of South Africa. So you can see here, as I looked at, we have the whole mitochondrial genome. So there's lots of stuff to choose from, and yet there's hard to find places that you can get just one species and get a good primer for them. I've created some for ND4. I looked at every gene in the mitochondrial genome and compared them to the number of changes they have and what percentage difference they are. Found ND4 to be the one that most likely will have possible probes. So I'm going to be hopefully testing that. So I should be getting the primer soon. If I can get some DNA, we can start working on testing those out. And then the yellowfish of the Sabi River. So there's 3 species we're kind of looking at. Just so you see this map here of Mpumalanga in the dark black here, I've kind of highlighted the main drainage of the Sabi River. So we have Nellis prudensis that's in colder water and should be found up by some of these amazingly beautiful waterfalls. If you've ever been up there, like Mac Falls and things like that, on the upper parts of that stream. But we're also looking at Polylepis and Marquensis. Marquensis should be kind of all over the place. And then Polylepis probably up higher, more like Nellis prudensis. So again, we've created primers, waiting for those to come in, got the DNA now for these things. And I'm looking forward to going out and collecting water up and down the Sabi and seeing if we can detect where different species and their species limits might be. And we'll see kind of how that goes. So those are kind of our major like species specific projects we're trying to do, but I'm also trying to work with trying to work with.
Hey, can you please mute?
Working with Tatenda and Gordon on some metabarcoding projects. So metabarcoding is a lot like regular just eDNA projects, but in this case, we can look at everything. So we collect a really thorough sample of water and a lot of it and extract as much DNA as we can, then amplify all the DNA in there and go through some high throughput sequencing, looking at CO1 and other markers. Nature Metrics has some really good assays for looking at different groups, and then they can give you the information on what species are found. Of course, this relies completely on there being data in the databases on the species. So what Tatenda and I are excited to do with one of his graduate students is we're going to collect a lot of invertebrates. Actually, they'll probably get to do all the collecting. I'll probably be doing a lot of the lab stuff. But they're going to sample in these pans that are in the southern part of Mpumalanga. So here's Mbambela here, Nelspruit, and then we went out the other day and found these great, really interesting pans full of all sorts of different invertebrates. And we're going to, they're going to collect, they're going to sample really, really well. We'll do some meta barcoding of that as well. And then we'll hopefully add some DNA to the DNA barcoding library for a lot of these African invertebrates. This student will help identify a lot of things. And if we can get funding, come to my lab in the States next year, the year after, where we can do some DNA barcoding and sequencing of these things and add to the database. And then last, but not least, I haven't started this project yet. I know Gordon has, but we're going to be doing a lot of meta barcoding up and down, doing some biodiversity surveying in the Limpopo River Basin to see what is there. They've got a big grant to kind of just scope out and look at this whole thing, and I'm just going to be one tiny little part of that, hopefully analyzing a lot of the data. And so looks like I'm pretty close within my time. Thank you so much for attending. I know I went really quick. If you have any questions about probes or protocols, we can talk about that now. You can email me at meg26@psu.edu. Happy to, love to collaborate or talk or, learn more from other people, whatever we can. So I guess with that, I'll turn it back over to Tenda for maybe some Q&A.
Thank you, Matthew, for the presentation. So now I turn to the people who have been listening. If anyone has any questions, they can write their question on the chat, or they can raise their hand, and then I will try to recognize them. Then I'll give them the floor to ask a specific question that they currently have for Matthew. Let's see, chats. Currently on the chat, there are no questions. Any questions from?
There's a question in the Q&A.
Okay, let me check. Okay, let me check the Q&A.
And then again in the chat.
Okay. Q&A. Anonymous. Okay. These are just students that are asking something related to the module that they are currently learning. All right.
Oh, good.
For the students, I will explain to them how the link exists. So I need questions that are more specific to the talk. Let me see chat. Okay, if there's a question, if we send samples from Malawi, how much will it cost to carry out the analysis? How about generating reference DNA for our fish species in Malawi? Any estimation of the cost? Matthew?
Ooh, that's a great question. So a lot of that depends on the species we're looking at. If there's species that are already known and they've already been sequenced, getting primers are pretty inexpensive. At that point, it's kind of the chemicals for running a PCR and running a gel to kind of see if you're testing for presence absence. I don't know how much, I guess I've never thought of it kind of from a business perspective, but if someone wanted to collaborate, that would be a really a fun project to do and to think of. If you have an idea of the organisms you're interested in, I'd be happy to look up to see if there's DNA data available, if there's a way to make probes that would work, and a way to kind of estimate, what is there and what might not be there. If you want to just send it away for meta barcoding, I know those samples are a little bit pricey. If you want to take like a water sample and just see what all the things that might be in there from one place, I think they quoted me, it's around $300 to $400 US to do one meta barcoding sample, but it is a lot of data that you get back through sequencing of that. So a lot of it depends on kind of the question you're asking, but if you wanted to email me maybe more details of the question, I'd be happy to kind of think about it some more and see what we can, what the range of your project would be and how it could get done. That'd be really interesting.
Thank you for that. I think the person, I think they will be in a position to email you, and then you can discuss further on how to go about the project. Any specific questions that you want answered in the Q&A? I'm not seeing anything. Let me check the chat. It's either it means that people understood the projects that you are currently the projects that you are currently working on, or you have left them confused. It's either the two, but I take the latter. It means that you understood. There's something popping up. Okay, this one is from Nick Nick Mandrick. So tigerfish is a good example of the challenge of cryptic speciation, particularly for meta-barcoding, and necessitating the need of tissues to develop primers. We recently published a Behrend River paper with Albert that outlines this challenge. I think it's more like I can say it's not a question, but more of a comment. So Nick, I think Nick is based in Canada. So I think he has done some work also within the Great Lakes, if I'm not mistaken.
Thank you. I will definitely check out that paper. I appreciate it. I just took note of it, so I'm gonna check it out because tigerfish are very new for me.
Okay, there is another question within the Q&A section. So variation in DNA, H. vitatus, is it about the confidence of the sequences that you are getting? In other words, how confident are you that the DNA sequence is a good sequence?
So that's a really good question. Right now, unfortunately, I haven't had a chance to sequence any of them yet. I'm hoping once we can get more samples and survey a lot more, we can kind of get a picture of what we have. It seems in this area, we're going to be focused kind of on this area and just a few river systems. So we probably won't get too much variation. You know, the Vitatus variation seems that it goes all over different, the continent of Africa. And so the variation in the populations is kind of spread out, seems to be spread out pretty far. But right now I haven't sequenced it yet. So I guess I don't have a great answer for how confident we'll be. I think once we get the working probes, we should be fairly confident that we could figure out if we're looking at the tigerfish that are here and not something else. We're mostly trying to delimitate them between other species of fish. Oh, I think you're muted, Tatenda.
Okay, yeah, thank you for answering the previous question. There is one from Pumza. She's asking, that did you notice any patterns in species distribution? And let me read one from Roger Bills. Other eDNA projects I read about use markers like 16S and 12S. Why are you using the C01? So there are two questions. The first one, did you notice any patterns in species distribution? Then the second one, it says eDNA projects use markers like 16S and 12S. And then why are you using C01?
Those both good questions. I haven't had a chance to do the widespread sampling that we're hoping to do on like the crayfish and possibly some mollusks and tigerfish. Been trying for the last few weeks to get a guard so we can go out in Kruger and sample a lot of the water and start really getting some of this going. So I don't have any species distribution patterns yet, but hopefully we'll be rolling and getting a lot of data in the next few months. And I'm sure I'll be taking a lot of DNA home with me to kind of work out even after I leave. As far as CO1, I've been sticking with CO1 because of it's kind of universally known as a DNA barcoding gene. And so there's typically a lot more data on that than other things. But if someone's done a recent phylogenetic project on 16S or 12S, then it would make a lot of sense to make a marker out of that, whatever you can get the most data for. It's nice to add data to the databases for CO1 because they have that as the kind of the barcode of life project. But I wrote a paper a few years ago on scale insects, and the most data we had was on 28S and 16S. No, it's 28S and I think it was EF1 alpha. I can't even remember. So we use that for actually detecting invasive scale insects and down in Florida. There wasn't good data on CO1 near as much as everything else. So kind of depends on what you're looking at. And I'm going to use ND4 probably for eels if I can make it work. Yeah, got to go with what you got.
Okay, thank you. There's a few more questions in the Q&A. So I'll start with the top one for the alien invasive species detection, where there is not sufficient variability or too much variability in the CO1 fragment. Have you considered a mini barcoding approach? This involves targeting a much smaller fragment of the CO1 gene, but I'm not sure if that will influence the qPCR signal. And so you can answer that, and then there are two more. Yeah.
Okay, yeah, I'll go with that. So that's a good question. When we actually use a pretty small piece of CO1 for Daphnia, we use it's about 162 bases. For Hemimysis, I want to say it's about 150 bases. For Nidolopsis, it's about 250 bases. So again, you're right. Whatever we can use to find good primers, we actually have been using pretty small segments, and they work really well in the qPCR assays that we've done. Work incredibly well, as we saw from Daphnia and Hemimysis. So the small pieces are great. Good comment.
Okay, cool. Then the next one, it says the eDNA, how is it used? Is it related to other methods for aquatic invader?
So I think if I understand the question right for that one, it would be that we're using it to try and locate where aquatic invaders are, and that's what we've used it in the Great Lakes. We use it to, Daphnia lumholtzi is really small, and the Great Lakes are massive, and so sampling all of them is going to be completely impossible. Sampling water is a lot more efficient and easy. It's still pretty hard on something the size of the Great Lakes, but if you can take a water sample and like Ivor's little catchphrase, evidence of presence is evidence of presence is presence of evidence. I'm going to get it backwards, but yes, it works. And so using that can help us detect the aquatic invaders sometimes more efficiently and quickly than trying to catch them, well, I'll just leave it at that.
Okay, and then the last one in the Q&A, how does the environment, e.g. temperature and salinity, affect the detection of eDNA?
So that's a good question too. We've had the experiment trying to determine how fast it's lost, and we actually tried a number of different variables in some of the experiments we set up, and temperature was one of them. Keeping tanks warmer or cooler. We did not find a huge difference in warmer versus cooler tanks, but to be fair, we were looking at our experiment was focused around the Great Lakes, so we were trying to mimic regular summer temperatures of lakes, which don't get overly hot. It's kind of northern part of North America. We didn't test really, really hot temperatures, which could drastically impact the amount of DNA. Our hypothesis was, the warmer the temperature, the more the microorganisms, the bacteria things would chew up and consume the DNA. And so that could potentially happen. and we have not had a chance to test salinity, how salinity might affect it. You have to kind of look at, what that does to DNA preservation in general, and it would mostly probably just be good. It might help us, lower the signal, really give you a good idea that it's there. But if it masks it completely, if it destroys all the DNA, I can't see it all destroying it all once it's being shed. So I don't have a good answer for salinity, but temperature shouldn't impact it too much.
Okay, thank you for that response. There are a few questions in the chat, so let me read those out. So it says, I apologize if you haven't mentioned this before, but can eDNA be used to identify species that previously lived in an area from stratified layers of the sediments at the bottom of the lake? All right.
Yes. I actually just saw a paper yesterday. I didn't have a chance to read it all. I think it was from Alan Cooper's lab in Australia, and I believe they were taking core sediment samples and looking at the changes of organisms over time. The biggest question is preservation. So I had a friend years ago, he was working on Daphnia, and they were looking at Daphnia eggs that were down in core sediments under Arctic lakes. So they kept in really, really cold temperatures, but those are full eggs and you can actually hatch them out. So ostensibly, if you took core things out of there, it might still be there. If it's just in water, likely it's going to be destroyed, but I don't know. I haven't done a lot with long-term preservation. I know there's lots of tricks people are using to get out DNA of things that have been dead for a very long time, but usually they have a pretty good sample of the thing and it's been cold for a long time. So I'd say it probably would have to be cold, and it might be whole organisms that are just down there being cold. That's a really cool question. I like that.
There is one more in the chat. How concerned are you that eDNA does not overestimate species variability?
That's a good question. I'm not sure exactly how to answer it. Typically, when we're using eDNA, we're mostly looking for presence, absence, and looking for distributions of things. Other labs are looking in different ways at eDNA, probably trying to get things like population genetic estimates, looking at different population structure. Ours, using qPCR and Agros, we couldn't actually really see much variation in the thing. We can see presence, absence. Is it there? Is it not there? We haven't done a lot of sequencing off of the eDNA yet. So that is a really kind of a cool question. And I'm sure it is very useful. I believe Sandy has done some population genetic stuff. I'm not sure if they've done it with eDNA or just with whole specimens. So probably could be done and be very cool, but I haven't done much sequencing with it. I think you're still muted, Tatenda.
Okay, sorry about that. Let me check if there's more questions in the Q&A. In the Q&A, there's nothing. Chat, I think there's one. Can you really use eDNA to assess the temporal distribution of a species? How effective is it in this regard? That came from Pule.
Yes, I think you definitely can, especially obviously with aquatics. If you're sampling your area well and you're finding presence of it, then sampling a whole area, you should be able to get a pretty good estimate as to where the organisms are. Though you have to be careful. You couldn't go to, say, a single dam and sample one place, and maybe 200 meters away, there's a little population of crayfish or something you're concerned about. Taking water from one place, you can't say, oh, it's definitely not in this dam because we took one sample and didn't find it. I think it's more of a, if you find it, then you can say it's there. you'd have to be really careful with the, with the negative, saying, oh, we didn't find it, it's not there. I think that's the biggest fear with it. If you're finding it right now, we've kind of confirmed if you're getting the eDNA sequence, or signal, you're getting the organism. You just would have to be careful to, to say it's not here after sampling one place on a big dam or something like that.
Let me check. Thank you for your response. Let me say Q&A. There is nothing. Check chat. There is no comments. Does the audience have any other questions that they want to ask Matthew?
Oh, another one popped up in the Q&A.
Okay, let me check. Yeah. How easy is it to cross-contaminate samples used for eDNA at various stages in the field, from genetic tissue collection to lab work, especially if you are looking in region? Or I think maybe the question wanted to say, especially if you are working in regions with high biodiversity instead of looking in a region. Yeah.
That's a great question, and that's a really important thing. You've got to be extremely careful because it's really easy to cross-contaminate, especially after you've run PCR. If you're doing the same gene, say you're doing lots and lots of samples, looking for the same organism in all the samples, the same gene, it could cross-contaminate your PCR really easily. So in all steps of this, you got to be very, very careful with your water collection, with your DNA extractions, and then with the PCR especially. That's one reason we went to a lot of the qPCR, one of the many reasons, but with the qPCRs, our number one rule in the lab was never open one. The thing reads it, and you look at it, get the data from the computer, and throw it away. Don't ever open the thing, because it just contamination. And once you've got it, it can be just a massive problem to get rid of. So yeah, and we actually did we set all the PCRs up in one room with a UV hood, and we ran them all in a different room. And the other rule was don't ever bring any of it back into the PCR lab. You got to be really careful, especially if you're making claims about invasive species and things like that. You got to make sure you're not contaminating anything.
Okay. Yeah. Okay, thank you for that. I think there is another question. It's not a question, it's just they're just highlighting that the recording will be uploaded to the SAIAB YouTube channel, and then there is a link. Any more questions? Okay. I'm not seeing any questions.
Okay. Can I
Okay.
say one more thing really quick?
Yes.
I apologize. I think I was on a lot of adrenaline right when I was starting. I meant to I really want to share my gratitude with NRF and SAIAB for this opportunity to present. It was a great honor to me as a researcher to be invited to this by Tatenda and by the group. And I'm, again, really grateful for Tatenda and Gordon and their labs and all that they've done to help me here. My time in South Africa seriously is the greatest place. I'm having an amazing time, and I love all the people I've interacted with, and I just can't get enough of it. So thank you very much.
Okay, thank you, Matthew. Okay, let me do a quick check before I say my concluding remarks. Let me say chat. Okay, there is nothing. So yeah, I would like to take this opportunity to thank Matthew for giving us the talk. And I would also like to take the opportunity for thanking the acting managing director for SAIAB and the chief scientist Albert Chakona for giving us the platform and for giving Matthew the platform to come and present. Thank you to all the members of the SAIAB staff that were involved in preparing this presentation. And thank you, Matthew, for honoring the invite. And yeah, we look forward to continuously working with you. And I hope that a few people will contact you via email talking about projects where you can collaborate and work with them. Thank you to all the participants for coming through to listen to the presentation. eDNA PA to ZA. Yeah, it was a fantastic presentation. With this, I say good morning and have a lovely a day ahead and a great holiday tomorrow. Thank you and good bye.
Thank you.